our last video, we talked about pulmonary ventilation, which again is just a fancy name for the movement of air into and out of the lungs. With this particular video, we're going to pick up where that left off. So we've talked about how air gets into the lungs. What I want to do now is talk a little bit about what happens to that air once it's actually in the lungs. So if you look at this diagram that we've got here, you'll notice that we have a single alveolus, one of those microscopic air chambers that's in the lungs. And you can also see the blood and the boundary between this alveolus and the blood. And you'll notice that we've got an arrow indicating that oxygen is moving from the alveolus into the blood, and another arrow indicating that carbon dioxide is moving from the blood into the alveolus. This is a process which is known as external respiration. So it's just the exchange of these respiratory gases between the lung and the blood. And this is driven by pressure gradients primarily. So the air that we breathe in that's sitting in the alveolus has a really high concentration of oxygen. For that reason, we're going to see oxygen moving from the alveolus into the blood. And at the same time, the blood has a really high concentration of carbon dioxide. The reason for that is this is venous blood. So it's been through the systemic circulation. It's given up most of its oxygen to the tissues of the body. At the same time, it picked up carbon dioxide from those same tissues because carbon dioxide is a waste product of cellular respiration. So we've got a high concentration of this carbon dioxide in the blood, and it's going to, because of that, diffuse into the alveolus so that we can breathe it out when we exhale. So we've talked about this pressure gradient that's driving oxygen into the blood. What I want to do now is talk a little bit about what happens to this oxygen once it's in the blood. So once it has moved from the lungs, from the alveolus into the bloodstream itself. Most of the oxygen that makes its way into the blood is transported bound to a chemical or a molecule which is known as hemoglobin. A little bit of the oxygen actually stays just in the blood, and it's just diffused in the plasma, and it just kind of floats along in the plasma. However, oxygen is not really very soluble in plasma, which is mostly water. So most of the oxygen does make its way into red blood cells, where it binds to this molecule which is known as hemoglobin. So we talked a little bit about hemoglobin back when we did the blood chapter, the first chapter in 228, and hopefully this picture, because of that, looks a little bit familiar to you. Hemoglobin is a molecule that's composed of 4 polypeptide chains. So if you look at this example that we have here, we've got one chain represented in kind of this tannish color. Here's a second one in purple, a third one in pink, and a fourth one in this dark brown color. A polypeptide chain basically is just a chain of amino acids. But what's important about these hemoglobin molecules is you'll notice right here in the center, we've got this little red representation of an iron atom. And that iron atom is where oxygen actually binds to the hemoglobin. So if you look at a single molecule of hemoglobin, there's 1, 2, 3, 4 sites where oxygen molecules can actually bind. And what that means is each hemoglobin molecule is capable of transporting 4 oxygen molecules that are attached to it. And one thing that's kind of interesting about hemoglobin and about red blood cells, there are about 250 million molecules of hemoglobin in a single red blood cell. Each of those 250 million molecules of hemoglobin are capable of binding to 4 oxygen molecules, and that means within a single red blood cell, we are able to bind about 1 billion oxygen molecules, which is a really incredible number. If you multiply that by the fact that you have trillions of these red blood cells in your body, our blood has an incredible ability to transport oxygen and a really high oxygen carrying capacity. So I want to talk a little bit more about oxygen once it gets into the blood and how it's actually transported by the hemoglobin. And we'll use this slide here to kind of just give you a verbal description of it and talk about some definitions. And then with the next slide, our last slide, we'll look at how this all relates to what's going on with oxygen levels in the blood. So what I want to talk about here is a process which is known as facilitation, and it has to do with how strongly hemoglobin is actually binding to the oxygen. If you look at this hemoglobin molecule that we've got represented for us here, again, you can see those 4 binding sites that are capable of binding to oxygen. And we've talked about the fact that each hemoglobin molecule, because of those 4 binding sites, can bind to 4 oxygen molecules. If oxygen levels are really high, so if we're talking about arterial blood that's fresh out of the lungs, it's likely that all 4 of those hemoglobin binding sites are actually bound to oxygen. But of course, you know, as the red blood cells, as the blood is making its way through the body into the systemic circulation, what happens is oxygen starts to break away from the hemoglobin, it diffuses out of the red blood cell into the plasma, and from there, from the plasma, it diffuses into tissues that need it for cellular respiration. What that means, in effect, is you can have a hemoglobin molecule that's saturated, meaning that all 4 of its binding sites are bound to oxygen. You can have a hemoglobin molecule in a red blood cell that has none of its binding sites actually bound to oxygen, or you can have a hemoglobin molecule that has some of its oxygen binding sites actually bound to oxygen, which in that case is known as a partially saturated hemoglobin molecule. The one thing that you should know about this, now that you know that a hemoglobin molecule can be bound to a single oxygen molecule, or 2, or 3, or 4, is when you have a hemoglobin molecule that's not bound to any oxygen. Binding of that first oxygen molecule to one of the binding sites is really difficult. Hemoglobin doesn't want to bind oxygen when it isn't already carrying any oxygen. This is what's known as facilitation. However, once it's bound a single oxygen molecule, it becomes easier to bind the second molecule, easier still to bind the third molecule, and very, very easy to bind that 4th oxygen molecule. The reason for this is each time it binds an oxygen molecule, it actually changes shape, which makes it easier to bind the next one, and easier to bind the next one, and easier to bind the next one. The flip side of that is when you have a hemoglobin molecule that's fully saturated. So again, that means that all 4 of those oxygen binding sites are actually bound to oxygen. We've got 4 of oxygen molecules that are attached. If that's the case, hemoglobin tends to hold on very stingily to the oxygen that it has. So if it's bound to 4 oxygen molecules, it does not want to give up the first one, and it holds on to it very tightly, and it very begrudgingly gives up that first oxygen molecule. Once it has given up one, that causes it to change shape, which makes it a little bit easier to give up the second one. Giving up that second one causes it to change shape again, which makes it a little bit easier to give up the third one. Giving up that third one makes it very, very easy to give up the 4th one. So the way that hemoglobin loads, where it doesn't want to take on oxygen unless it already has some, and the way that it unloads, that it doesn't want to give up oxygen unless it's already given up some, is what's known as facilitation. And the big picture is when there's a lot of oxygen in the blood and there's a lot of oxygen in the red blood cells, hemoglobin is really stingy and it holds on to that oxygen really tightly. And the reason for this would be if there's a lot of oxygen in the blood and there's a lot of oxygen in the red blood cells, there's no reason for it to be giving up its oxygen, because that means that the tissues have what they need. They've got plenty. If we start to see oxygen breaking away from the hemoglobin, again, having that first one leave is really difficult, but that makes it easier for the second one, easier for the third one, easier for the 4th one even still. This is because hemoglobin is very generous with its oxygen and it gives it up very readily when there's very little oxygen in the blood plasma. And the reason for this, of course, is if there's little oxygen in the blood plasma, we certainly shouldn't have oxygen hanging out in the red blood cells where it can't be used by the tissues. Instead, we need to give that oxygen to the plasma where it can then diffuse into the tissues of the body. So that's the process of facilitation. Again, just describing kind of how hemoglobin binds to oxygen and the fact that that will change based on how much oxygen is in the plasma. This last slide is a diagram that kind of shows this relationship in a mathematical form. So because of facilitation, we end up with a situation where the relationship between the blood plasma, oxygen, and hemoglobin saturation isn't linear. So if you look at this diagram over here, I want to walk you through this a little bit. Down here on this axis, on the x-axis, this is our representation of the amount of oxygen that's in the plasma. So this is the PO2 of the plasma. So in the blood, how much oxygen is dissolved in the blood. Over here on the y-axis, we've got the percent of hemoglobin binding sites that are actually bound to oxygen. And what I want you to notice is over here, this would be the situation for arterial blood. We've got 100 millimeters of mercury, that's our pressure, or that's our concentration, that's our amount of oxygen in the blood plasma in arteries that are fresh out of the lungs. And when that's the case, if you look at this little blue line here, our saturation of binding sites in the red blood cells is about 98–99%. Where facilitation comes in has to do with the curve in this graph. So we don't have a linear relationship. We do not have the situation where each time we get a drop in plasma, we get an equal drop in hemoglobin saturation rate. That just isn't the case. Instead, what can happen is we get this relationship here where we've got this curved line. And basically what this is saying is, if we look at this end of our graph, this is times when we have a situation where the plasma has a high concentration of oxygen in it. And when there's a high concentration of oxygen in the plasma, there is no reason for hemoglobin to be giving up its oxygen, and instead it holds on really tightly to the oxygen, and it's very hard to break away that first oxygen molecule from the hemoglobin. If you look at this, you'll notice you can have a drop from 100 to 50. So we can basically half the amount of oxygen that's in the plasma and only get a 15% drop in the percentage of sites in the red blood cells that are actually bound to oxygen. So when oxygen levels in the plasma are relatively high, hemoglobin holds on to oxygen very tightly. It's very stingy. It doesn't give up its oxygen. And because of that, the oxygen saturation rate, or the percentage of hemoglobin sites that are actually bound to oxygen, are very, very high. If you look at this other side of the graph, so if we look at this portion of it here, this is what's happening in the blood when oxygen levels are quite low in the plasma. So when there's not a lot of oxygen flowing around in the blood plasma, that means there's not a lot of oxygen to give up to the tissues of the body that need that oxygen. The whole role of the blood is to pick up oxygen, distribute it to cells that need it, and pick up carbon dioxide as well. So if we have low levels of oxygen in the plasma, that's a problem. And when that's the case, what we're going to see happening is hemoglobin is not going to be so stingy with its oxygen, and it's going to start giving up very rapidly. And we're going to see on this end of the graph where our plasma levels of oxygen are quite low, that hemoglobin is very willing. It becomes less stingy in that situation and very rapidly gives up oxygen molecules, which makes the saturation rate in the red blood cells decrease really rapidly, as you can see on this side of the diagram. But that makes sense because if we've got low levels of oxygen in the plasma, that means we don't have enough oxygen to give up to the tissues of the body. That means it makes no sense for our red blood cells, for our hemoglobin to be holding on to that oxygen and being stingy with it. Instead, we at that point need the hemoglobin to start flushing lots and lots of oxygen into the plasma so that that oxygen can be given up to the tissues of the body.